Hi students. So myself Alexander, biology teacher. If you like my channel, please press the like button and share with your friends. And today I am going to discuss about the topic that is in cell, the unit of life. It's taken from the 11th CBSE unit 3. Okay. So cell, the unit of life. Have you visited any construction site? If you have visited any building construction site, what do you observe in that place? What do you observe in that place? Are they making the walls with the help of the bricks? Making the foundation? Have you seen the wall, the brick wall? So line by line, layer by layers, they are arranged and packed tightly to form the wall. Am I right? The same way in our living system also. The organ, tissue, all are made up of a group of cells. These cells are arranged and tightly packed. The cells are arranged, tightly packed or loosely packed, but they form a tissue. Okay, so that's why we call it the cell, the unit of life. The unit of life. To make an organism, how the cell is playing a major role. A group of cells, the form tissue, group of tissue forms organs, group of organs form organ system. The group of organ system put together forms organism. So what is the function of the cell? How this cell is considered the unit of life? For the basic formation of the organism, which is needed, the basic one is cell. Cell is independent existence. It's able to do its different activity to make the organism is alive. The cell is not doing the functions, then the organism having the defect. So the cell is doing a function, different activities. That's the activities we call it an life process. Respiration, digestion, excretion, movement, all these things we call it in a life process. Okay, for the life process, for the essential function of the life, the cell is playing a major role. To do the essential function of the life, the cell is working out. That's why we call it in a cell is the structural and functional unit of life, unit of organism. It's the basic unit of the organism, the smallest unit of the organism, which are constructing the structures as well as doing the function, making the organism alive. That's why we call it in the cell, the unit of life. So Robert Hooke, the first person who has observed the cell, okay, he has seen like a compartment. So followed by him, Anton von Leeuwenhoek is a person who has seen the live cell, observed the live cells. Okay, later on, the Robert Hooke is discovered the nucleus. Okay, so the microscope with the revolution and evolution of the microscopes and brought light into the internal structure of the cell, structural components of the cell. So that's from the ordinary microscope to the TEM, transmission electron microscopes, TEM, SEM. Okay, all these the evolutionary microscopes has given the idea, has giving more knowledge about the cells in the modern society. Okay, so in 1839, the two scientists, okay, Sheldon and Swan, these two scientists proposed the hypothesis. What is this hypothesis? They proposed certain fact. The fact is all the organism is composed of one or more cells. The organism is composed of one or more cells. Okay, so the the organization of the organisms need the basic unit that is called cell. That is called cell. So in 1855, Rudolf, okay, so Rudolf Virchow, he has composed this hypothesis and brought a finalized statement of the cell theory. So what is cell theory says? All the organism composed of one or more cells. That's the first point of the cell theory. The second point is these organisms having the basic unit that is called cell. The third point is all the cells, the new cells, are formed from the pre-existing cell. Already certain cells are there. From that it divides and forms a new cell. So that is it. So cells undergo the divisions and forms a new cells. So new cells are formed from the old one. That is only the concept. All the cells are formed from the pre-existing cells. So this is, we call it a cell theory. So all the organisms are composed of one or more cells and their products. All the organisms having the basic unit, that is, we call it cell. Then all the cells are formed from the pre-existing cell. This is the cell theory proposed by Rudolf Virchow after composing the hypothesis of Sheldon and Schwann's theory. Clear? The cell size may vary and shape also vary. Okay, the cell size and shape. The cell size is not having any role with the organisms. That elephant is bigger, so thereby the size of the cell should be bigger enough. It's not like that. Regardless of that organism size, the cells are doing its fundamental activity. The shape of the cells may play a role because the shape of the cells are regarded with the position, its position as well as the function. What function it will do, what function it performs, based on that the shape of the cells may be varying. Okay, the smallest cells, we call it the smallest cell is an PPLO, pneumonia like organism. The size of this PPLO is 0.1 micrometer in length, whereas the bacteria will be having 3 to 5 micrometer in length. Okay, it is known as PPLO. It is also known as a mycoplasma. It is considered as a mycoplasma calcitoceptum. Okay, the largest cell, the biggest one, we call it an ostrich egg. It's 6 inches in diameter with the shell. Without the shell, then it is 3 inches with the diameters. Got it? So in case of multicellular organism, example, human blood cell. The human blood cells 7 micrometer in diameter. 7 micrometer in diameter. Okay, then the longest cell is a nerve cell. Okay, the longest cell is the nerve cells. The shape of the cells may be oval shape, a spherical shape, or polygonal shape, or elongated shape. Okay, a round shape, or biconcave shape, or amoeboidal shape. How this shape may vary, why it varies from one cell to another cell? What is the need? The consider RBC, red blood cells. The size of the shape of the RBC is round or biconcave. Okay, either it will be round or biconcave. But in case of WBC will not have the definite shape, so they are called as amoeboid shape. Amoeboid shape. Since that RBC is having the definite shape, it can't enter to the nook and corner of the body cells. It is possible. But this WBC is not having the proper shape, so thereby it enter into the nook and corner of our body cells, where it is not, where other cells are not able to enter it. Why it is able to enter into the nook and corner of the cells where the other cells are not able to enter? The reason is it's having the amoeboid shape. So it can change its shape according to the organs, according to the tissues where it enters. Finally, it reaches the destinations to work out, to function. Because this WBC function is to protect the body from the pathogens, to protect from the foreign substances. Okay, that is the function. That's why it is not having the proper shape. So wherever it is needed, it will easily able to penetrate, seep into that. Okay, then enter to the destinations of the particular site, target site. Okay, fine. So that is the role of the shape of the cells. The shape of the cells is regards with their position as well as the function what they perform. So let's move on to the next topic, that is based on the nuclear arrangement. Based on the nuclear, on the basis of nuclear arrangement, on the basis of nuclear arrangement, the cells are grouped into two. Number one is in prokaryotes. Number two group is in eukaryotes. Prokaryotes and eukaryotes. These words are derived from the Greek language. Okay, isn't Greek word pro means before. Pro means before. Okay, so karyon. Karyon means nucleus. In certain cells, the nucleus is not well defined. They do not have the nuclear bound membrane organelles. Nuclear bound membrane organelles, and the nuclear genetic materials will be infused in certain cells. Okay, they are the basic cells, so they are known as the prokaryotes. So prokaryotes, the membrane bound organelles are absent. The nucleus will not show the clear membrane, nuclear membrane, nucleolus, and nucleoplasm, then that genetic material. So it is indifferentiate. But in case of certain cells, eukaryotic cells, they have the proper and definite nucleus structures. They are having the membrane bound organelles, nuclear membrane bound organelles. So they are able to easily identify it. Okay, fine. So their genetic materials are also condensed and clear. Condensed and clear in eukaryotic cells. Got it? So in prokaryotic, the nuclear membranes are not clear. So nuclear bound membrane organelles are absent. Got it? Clear. That's good. So we will discuss the prokaryotes with one examples. And the best examples is the bacterial cells. So what are the things can come under the prokaryotes? Bacteria, we call it a bacteria. Bacteria comes under the prokaryotes. Chlamydia comes under the prokaryotes. Rickettsia comes under the prokaryotes. Blue-green algae comes under the prokaryotes. Okay, but what comes under the eukaryotes? So in eukaryotes, higher plants and animals comes under the eukaryotes. Plants and animals comes under the eukaryotes. Got it? So let's have. So I am drawing the rough diagram of the bacterial cell. Ratik Salman. Okay, let's label the diagram. Okay, so this is an mesosome. So you will identify calyx, the outermost layer. Sometimes it is called a capsule or slime layer. Okay, fine. This is ribosome. This is a circular DNA duplex called circular DNA duplex. This one is a membrane, plasma membrane. Okay, we'll be having this is we call it an flagella. Okay, this is basal, and this one is a hook, and this is an filament. Got it? So here we will be having the, this is we call it an glycogen granules. Okay, and we will be having the polyribosome. Got it? And we will be having the respiratory enzymes. Respiratory enzyme. Clear? Then we will be having the fimbriae, thin hair-like structures. We call it fimbriae, which is for the attachment. So it helps to attach the bacteria to the solid surfaces. The fimbriae. This is an fimbriae. This one is a fimbriae. Alright. So this is the rough diagram of the bacterial cell. Look at that. That bacterial cell is having the cell envelope. Okay. So three layers are tightly bound, forms three layered envelopes. Three layers are tightly bound to each other, forms envelope structure. That's we call it in cell envelopes. What will be the three layer? The outermost layer. Outer one is an glycocalyx. Outermost layer is in glycocalyx. Then we are having the cell wall, followed by the then you will be having membrane, plasma membrane. These three layers are tightly bound to form a three-layer structure known as a cell envelope. Okay. Then after that we are able to see the next layer on the surface of the cell wall. We are able to see the structures called the structure called glycocalyx. On the surface of the cell wall, there is a gelatinous surface layer. Gelatinous surface layer, mucilages, that forms on the surface of the cell wall. That layer is known as a glycocalyx. This glycocalyx may be tightly packed. If it is fused with the surface of the cell, then it forms a capsule. If it is not properly packed, loosely packed, then it forms a slime layer. Got it? Clear? So, followed by the cell wall. Then we are having the cell membrane. In the cell membrane, you are able to see that extension of the cell membrane forms tubular-like structure as vesicle-like structure. Okay, or lamella forms. They are known as mesosome. They are known as mesosome. Okay, the mesosome is playing a major role in the bacterial cell. What will be the major role of the mesosome? It is involved in the DNA replication. The mesosome involved in the DNA replication and distribution to the daughter cells. Not only distribution to the daughter cells, and it also involved in the cell wall formation. Mesosomes involved in the cell wall formation, and it is also doing certain activities like respiration, secretion, and protections. All these functions are done by the mesosome. It's a specialized structure which arises extension of the membrane. Plasma membrane forms the mesosomes and involved in the different activities. Then you are able to see the circular coiled DNA. It is a single circular DNA duplex. Okay, this single circular DNA duplex. Along with that, we are able to see a specialized structure called ribosome, which is involved for the protein synthesis. It is the site of protein synthesis, right? Which is site of protein synthesis? Ribosome. Okay, clear? Right. Then we are able to see that a number of ribosomes attached with the mRNA. That is known as a polysome. That's known as a polysome. Okay. Then we are able to see certain granules attached to the surface of the plasma membrane. That's we call it in respiratory enzymes. Okay. Then we are able to see arising from the plasma membrane, there is a basal body. Then we are having the hoop. Then followed by then we are having the filament. So these structures we call it a flagella. This is together we call it a flagella. Okay, and it's then like the same flagella, there will be another tubular but short structures. Okay, extending from the plasma membrane coming out. That's known as an pilus. That's we call it an pilus. So pilus is involved in the conjugations. When the one bacteria having sexual contact with another bacteria, there will be bridge formed with the help of the pile. Okay, this pile will open the bridge and attach with the host organisms, then transfer the genetic material through that bridge. Then the genetic material from the one bacteria to another bacteria is transferred. Okay, so analogous to the sex organ. Analogous to the sex organs. Clear. Another one is a very thin and long tubular structure. That's we call it a fimbriae. The fimbriae is helping to attach the bacteria to the solid surfaces. Okay, so we will go on one by one. So let's look at the cell wall of the bacteria. The cell wall of the bacteria is composed of peptidoglycan. Peptidoglycan. The cell wall of the bacteria is composed of peptidoglycan. We call it as an murate or muco. Mucosaccharides. Okay. So a bacterial cell wall is made up of number of layers of peptidoglycan. Based on the composition of the peptidoglycan, then the bacteria are grouped into two categories. One is in gram positive, gram negative. Gram positive bacteria, gram negative bacteria. So in case of gram positive bacteria, they respond the composition of cell wall. So very thick cell wall, many layers of the peptidoglycans. Because of many layers of peptidoglycans, the cell wall became very thick. Okay. But in case of gram negative, it is very thin because a few numbers of peptidoglycan layers are present. Okay, so that's why it's very thin. And when we see the gram positive bacteria, this type of bacteria, they respond to the staining process, gram staining procedure. Okay, so that's why when it gives a response to the gram staining procedure, it is known as a gram positive bacteria. If it is not giving that positive result, then it is a gram negative bacteria. So gram positive bacteria cell wall is made up of many layers of peptidoglycan along with the teconic acid. Okay, but in case of the gram negative bacteria, it is made up of a few layers of peptidoglycans along with the secondary polysaccharide. Okay, so that's the things regarding the gram positive and gram negative bacteria. So the bacterial cell wall is made up of peptidoglycan. Let's see what about in case of fungi. In fungi, cell wall is composed of the structure called chitin. Okay, specialized cellulose type chitin. In case of algae, so in case of algae, cell wall of algae is made up of glycoprotein and polysaccharide. This is algae. So cell wall of algae is made up of glycoprotein and polysaccharide. Okay, cell wall of plant, polysaccharide, glycoprotein, minerals. Okay, all these things forms the cell wall of the plant. So cellulose. Got it? So the cell wall is further is classified into three. Means it is having the three layers. Cell wall is having three layers. One is we call it an primary cell wall, secondary cell wall, middle lamella. Okay, in the primary cell wall, when the young plant cell which is having the primary cell wall, so it diminishes as it grows into the mature plants. Okay, so diminishes in the sense it is disintegrated, diffuse, and forms a secondary cell wall from the young plant to the mature plants, mature tree. So the primary cell wall is diminishes into and secondary cell wall. Okay, but middle lamella will be present and it acts as a transverse, and this middle lamella is composed of calcium pectate. So this middle lamella is formed of calcium pectate and it is present in the leaf, okay, leaf of the plant. and it is present in the transversal sections between the primary cell wall and the secondary cell wall, middle lamella. Okay, got it? Clear. So what we discussed in the bacterial cell wall? Bacterial cell wall is made up of polypeptidoglycans. So in case of fungi, the cell wall is made up of chitins, and in case of algae, the cell wall is glycoprotein and polysaccharides. In case of plant, polysaccharide, glycoprotein, and along with the water and minerals, all these things forms then the plant cell wall. Got it? Clear? Fine. So we will discuss the further the eukaryotic cells, and in that what are the cell organelles are present, and the role of the cell organelles and their function, and along with their structures. Thank you.